Hey guys, we are in chapter 8, genetics. This video is hopefully, I'm sorry about the wind chimes in the background, I really can't stop them. This video is to finish up chapter 8. Hopefully we'll get through all of looking at prokaryotic gene exchange, looking at mutations and repair mechanisms. I think that's it. So let's go over to the slides and we'll work through these. All right, for my class B440, this is the next set of slides, Genetics 2. As mentioned, we'll be discussing gene regulation in prokaryotic cells, what mutations look like and the consequences of mutations on the protein product, and then looking at recombination events and DNA repair mechanisms. So let's get started with this. Oops. There are some key concepts for this one that you'll see throughout the rest of the semester, essentially on this exam and the last one. Or the last two, depending on how the semester goes. The key concepts for this set of slides are the lac operon. Fortunately, we've already talked about this a little bit in chapter 7 when we're looking at enzyme regulation. We'll look at that in a lot more detail here, and that would be one thing that I do want you guys to understand the ins and outs of, or at least to the level that your textbook has this. The consequences of mutations on proteins is important, understanding how mutations affect the downstream polypeptide and therefore protein product, and then understanding the F factor or the genetic recombination, specifically in the conjugation. So overall, just consider prokaryotic cells. They are asexually reproducing organisms. Therefore, all offspring are genetic clones, essentially, of the parental organisms, which limits genetic variation, right? Essentially, asexually reproducing organisms all look exactly the same, or essentially they are all clones of each other. In fact, if you take each colony that you streak for isolation, when you streak for isolation in all of those individual colonies, all of them should look genetically identical with very little exception. The exception would be, I think I mentioned this in the last video, at every time there is a DNA replication event, every DNA polymerase, and you can look this information up, every polymerase, prokaryotic and eukaryotic, has inherent error rates. So there are always errors that are incorporated during a replication event. Most of the time those errors aren't meaningful in the grand scheme of things. They just have minor alterations to like non-coding regions or potentially regulatory regions on there. But when those changes occur in the coding regions of the DNA, then you can start to see some pretty significant impacts of these mutational events. But nonetheless, other than those minor mutational events, of those single nucleotide polymorphisms is what we call them, the genetic information is essentially exactly the same. So the problem is, of course, just imagine our classroom. We all have significant variations to our genetic information, and actually the variations are exactly what I described, those little single nucleotide mutations, or we call them polymorphisms, that are between you and me. Most of them occur in non-coding regions, but some occur in the coding regions and give us our variability, like phenotypically, the stuff that you actually see, but also metabolically, like differences in the ways that our bodies process different types of nutrients. Immunologically is very important. This gives us our variations and susceptibilities to particular pathogens, for example. So consider, though, let's just look at that last example, that all of us were genetically identical. So perhaps in our current day and age, now we have the ability to clone organisms. Maybe you decided with your multi-billion-dollar inheritance from your great-grandfather that you didn't know existed, that you had the ability to genetically clone yourself and you just made a whole room full of whatever's of Jeffs or something. So now you have all of these individual people with all the exact same genetic makeup, but that also means on the immunological level, all the same susceptibilities to the same pathogens, right? There's no difference from one person to the other. So let's say that Jeff happened to be particularly susceptible to COVID-19, this new strain that's going around due to mutations in your ACE2 receptor. Well, unfortunately, all of the clones in the room have the exact same genetic susceptibility. So when COVID-19 comes in and infects one of those individuals, everyone will get sick with that and die, right? And so now there's no more population, there's no more Jeffs, there's no more human race. So again, going back to our practical application for prokaryotic cells, this is the same concept. If an individual bacteria develops into an entire colony, that means all organisms within that colony are genetically identical. Well, every organism in that colony is now susceptible to the exact same whatever it is, environmental fluctuations, viruses, bacteriophages, right? Whatever that, that environmental change might be, all of them have equal susceptibility. So evolutionarily, organisms have evolved ways, even asexually reproducing organisms, to have genetic variation so that the population or the species can survive environmental changes. So these are ways that prokaryotic cells do gain genetic variation. Even asexually reproducing clonal populations have genetic variability. And so here are two different mechanisms. One, just inherent in a single prokaryotic cell. That organism can turn on and off various genes or various operons to respond to altering environmental changes, right? So we talked about this already. For example, when the key sugar that's used, often glucose, is gone, if there is another sugar present, the organism can change its gene expression so that it can now begin to metabolize that available sugar to help it survive in that varying altering environment. Otherwise, there are actual changes genetically that can occur within that population. I've already mentioned one, which would be mutations. So there are mutational events that occur during the process of DNA replication. That's natural, that happens all the time. There are also spontaneous mutations that can occur from environmental fluctuations. Those are unnatural, but still may confer some benefit. Those mutations occur, let's just go with that first example, during the process of DNA replication, which means from parent to daughter. So that would be a vertical gene transfer. Alternately, there are various types of recombination events. Recombination is horizontal gene transfer in that population from one organism to another existing within that same time period. These would be conjugation, so the actual connection of two bacterial cells through that sex pilus and the exchange of plasmids. Using bacteriophages themselves, well, I wouldn't say using, but sometimes that can occur, and that's transduction. Or transformation, just the naked DNA from the environment being taken up by competent cells. We'll talk about all of those different types of recombination events that can occur with prokaryotic cells. Here's a picture of these two concepts, vertical and horizontal gene transfer. This first one on the top, vertical gene transfer. In this case, an organism received a spontaneous mutation. In this example, that mutation occurred perhaps through environmental factors. And that mutation is then propagated in the population through replication events, right? This mutation is not going to be passed from one cell to the other, but when this one bacterial cell replicates, now it is incorporated in the population because daughter cells will propagate this essentially during every replication event. So this is vertical transfer, where that mutation or that genetic variation is transferred or passed in the population, not cell to cell, but during a replication event. Down at the bottom is horizontal gene transfer. In this case, we're looking at something like transformation with naked DNA from the environment being taken up by a neighboring cell, or perhaps if this is a direct connection with that sex pilus, this would be conjugation, an example of gene transfer from one cell to the other. Both of these would be horizontal from one bacterial cell to another within that population, but then note this will always lead to additional transfer as that one altered cell replicates and passes on that newly acquired genetic alteration to its offspring. So horizontal is across, of course, and vertical is up and down. Let's look at how a cell will regulate its gene expression first, and then we'll go back and talk about those mutations and what their consequences are on protein products. So I know we've talked about this in chapter 7, that organisms can control the expression of various types of genes that encode for various types of proteins or enzymes in particular. And so these are the control mechanisms that they have in place to turn on or off genes. And for prokaryotic cells, this is especially important because they really have no way to store energy, right? So whatever they have available to them is essentially what they will be using. and they need to constantly acquire nutrients from the environment to produce more energy. So in order to conserve their energy and not waste it, they will only express genes encoding for enzymes that are actively needed at any given time, right? So genes are active only when the products are needed, or of course, when the substrate is present. For prokaryotic cells, we've talked about these concepts of operons. So an operon is a collection of related genes, right? We talked about this for lactose catabolism or other catabolism units, where all of the genes encoding for all of the enzymes involved in a single metabolic pathway are regulated together. And this makes sense, right? If a pathway is needed, it will be turned on, or the genes will be expressed into enzymes. When the pathway is not needed, then those genes altogether can be turned off, essentially with just one on-off switch, instead of having to regulate each individual gene separately. Eukaryotic cells, we don't do this same thing. We have many-fold regulation components to many of our genes. It gets kind of complicated there, but for prokaryotic cells, they have these operons regulated as a single unit. We've talked about these operons as being either inducible or repressible based on whether the genes, the enzymes encoded by those genes, are involved in a catabolic reaction or an anabolic reaction. And let's just remind ourselves what these guys mean from our last chapter. For catabolic reactions, if I can get my pen up here. For catabolic reactions, these ones do what? Break bonds or form bonds? These ones break bonds. Right, this is often where nutrients are brought in for the production of energy. Using those central metabolic pathways we talked about in our last chapter, versus anabolic. Operons or anabolic pathways are also termed biosynthesis, or synthesizing larger components, macromolecules, into cellular components in a prokaryotic cell. And these ones require the input of energy. I'm sorry, I don't know why it does that sometimes. The input of energy, right? So if an operon is involved in breaking bonds or producing energy, these catabolic reactions, these guys will be turned off. So inducible means normally off, can be turned on, right? Normally in the opposition, can be induced or turned on. And the opposite would be true of these, I'm gonna move things around a little bit so I got more space, of these repressible operons. These guys are normally expressed always, so they're normally in the on position. You usually have gene expression of these biosynthesis things. And remember, we talked about when that product is not needed in the cell, it can come back and turn off its own synthesis. We call that negative feedback regulation, if you recall. So these can be turned off. Through negative feedback. Negative feedback regulation. All right, so let's look at these in a little bit more detail. Inducible operons and repressible operons. We're going to start with inducible operons, in particular the lac operon. The lac operon has some very important units to it. So a regulatory unit that's down or upstream, somewhere that's not connected to this particular operon unit. And the regulator, the regulator is a gene that encodes for a repressor. The repressor molecule is an enzyme that has an active site that binds to the operator region of the lac operon, and it has an allosteric site that is involved in regulation. So this enzyme, this repressor enzyme, functions just like what we had talked about in chapter 7. The repressor is actually an enzyme. So it has, again, an allosteric site involved in regulating indirect regulation of this molecule through allosteric regulation, and an active site here that binds to the operator unit. Whoops, sorry, it looks like I skipped a slide. Okay, That's the regulator. The control locus is here at the front of this, the actual encoding region of these of this operon, where the genes are. The encoding region has a promoter unit and an operator unit. And if you recall from our last video, we talked about promoters as being a sequence of nucleic acid where RNA polymerases will bind. And then this operator unit, I talked about this also in our last video. The operator is the site of regulation for the expression of these genes here. And so this now I can show you is where that repressor unit will actually physically sit and dock to prevent the expression of everything here. And then there is the structural locus or structural region of this operon, and this is where the actual genes are. And for the lac operon, there are 3 genes involved in this process. Let's go over and look at this in a little bit more detail. Okay. So we're going to start up here by drawing out the lac operon. Again, what we're talking about here, the lac operon is an operon or collection of genes involved in regulating the catabolism of this particular sugar, lactose. So the lac operon is involved in lactose catabolism. Lactose is a sugar, right, a carbohydrate. This is a catabolic operon or catabolic pathway, and therefore this is usually repressed, and we can call this an inducible operon, or it is normally repressed and can be induced. Okay, so again, we have our key regions here. Here's our double-stranded DNA molecule. We have up here, somewhere upstream, is the gene, which is called the lac, what do we call this, the lac-Z gene? Lac-I? Let's go check that out. Lac-I, that's what it is, sorry. Okay, this is called the lac-I gene. And the lac-I gene encodes for the repressor. Okay, let's draw that one in. So we have our repressor, and I'll draw that one in green. The lac-I gene is transcribed and translated into a repressor. I'm going to draw this just like our last enzymes here. This is the repressor. And just so you're remembering, we have an active site and an allosteric site. The active site on this repressor, the active site here, for the repressor is the DNA at the operator region of the DNA. So let's draw our actual operon here. Here we have the operator, O for operator, which is like our on-off switch kind of thing, right? This is the regulatory site of this operon. We also, on our operon, have our promoter region. Sorry if the colors are off from before, right? This is our promoter, P, versus this red guy. This guy here is our O. Let's draw O up here on the top so I can do my regulator on the bottom. All right, so the promoter is where we have our RNA polymerase binding site. Okay, RNA polymerase binds here, or docks here. So a couple of things to note here as we're drawing this out. Oops, let me also add in, sorry. Let's add in our structural genes down here. So we have gene 1, gene 2, and gene 3. Now, just so you're aware, okay, these genes are responsible for, these genes encode for enzymes responsible for bringing in lactose into the cell and for catabolizing lactose into usable glucose. Let me just show you a real brief picture of how that looks. So when lactose comes into the cell, if you recall, this is our cell. Lactose, and I'm going to draw lactose in pink. Lactose has to first get into the cell, right? And this part actually requires a transport protein called a lactose permease. And actually, gene 1 encodes for the lactose permease. Okay, so that would be gene 1. Oh, let me draw that in more regular colors. Gene 1. Now I will say that lactose permease is this guy right here. This regulation is not completely efficient, so there's always just a very small amount of these genes being expressed at all times, even with repressed operons. So there's always a tiny amount of this lactose permease present, which allows a small amount of lactose to get into the cell, even when it's not being used. So lactose gets into the cell, and it needs to be converted essentially into usable sugars. So lactose needs to be broken apart. This is a disaccharide. It's broken apart into glucose and galactose. These are the two sugars that make up this molecule. And this enzyme here, this enzyme would be gene 2's enzyme, right? Or enzyme 2. This is called beta-galactosidase. Encodes, so gene 2 encodes for the enzyme that does this reaction. And then that last step would be this galactose needs to be further modified. And so this is where gene 3 comes in. Right? So here's our 3 genes involved in the lac operon. Gets lactose inside, breaks it apart into glucose and galactose, and further modifies galactose to usable sugars. So these guys are usually suppressed. There's usually almost nothing expressed except for a tiny, tiny amount to allow lactose to seep in its very, very small quantities. So when lactose is present, though, all of these genes need to be turned on to make these enzymes involved in this reaction, right? And remember, I'm putting gene, I'm hoping that you remember genes aren't actually the things that are doing this. It's the enzymes that are synthesized from these encoding regions of the DNA that are actually doing each of these steps here. But just so you see where these things fit in. So again, normally these genes are in the off position, they're not being expressed. And that's for 2 reasons. One, your book doesn't talk about this very much, but there is, again, this is, I'll just put this giant thing here as the RNA polymerase. So the RNA polymerase will bind and dock right here at the promoter region, and it needs to go in this direction, of course, to begin to transcribe these genes and translate them. So one thing that occurs is if there is glucose in the cell, glucose will prevent essentially the RNA polymerase from binding here. So glucose, the presence of glucose will prevent this whole thing from happening. So just so you're aware, again, your book doesn't talk about this in detail, but if you watch any of the videos, you'll hear something about cyclic AMP needing to be present in order for RNA polymerase to come and bind. We did talk about AMP, if you recall, adenosine monophosphate. as being a trigger to the cell of cell death because there's not enough ATP being made, and so ADP is actually dephosphorylated to produce energy to make AMP. So AMP becomes a signal to the cell that there's essentially no glucose present or no energy being made, no catabolism happening. So that is the trigger to turn this whole thing on. But we're going to keep it simple. You don't have to worry about any of that stuff, and just say when glucose is present, essentially when it's being broken down to produce energy, then its presence in an indirect way will inhibit the binding of RNA polymerase. RNA polymerase cannot bind. When glucose is absent, RNA polymerase will bind. Okay, so there's one step right here. The second factor, and the reason that we're actually talking about this as an inducible operon, has to do with this repressor. The repressor, as I just mentioned, has the active site as the operator. So the repressor will always be sitting right here, right? It will be docked here, essentially at this region. This is the repressor. And when it's docked here, then even if RNA polymerase comes and sits down on this promoter region, this repressor will completely block the RNA polymerase from moving forward for transcription, right? So this repressor blocks here. So here's two different ways that this operon is inhibited or repressed. One is that the presence of glucose prevents the RNA polymerase from binding in the first place. But two, even if glucose is absent and RNA polymerase can bind, the presence of the repressor on this operator region prevents the synthesis, the transcription of this operon, because it physically blocks the polymerase's access to the DNA. Okay, so let's just focus on this right now. As mentioned, there is an allosteric site, right, on this allosteric site here. Allosteric site. And if you recall, this allosteric site binds an allosteric regulator. In most cases, this is so this is an inducible operon, and we had talked about how for inducible operons or catabolic operons, the substrate of this metabolic pathway will be the inducer. And actually, because it acts as an allosteric regulator to the repressor. I know that's like two different levels to think about, but the allosteric site, in this case, is where we will have the allosteric regulator come and bind to the repressor to make the repressor not work anymore. So this is where something called allolactose, it's a derivation from lactose, but we're not going to talk about that here either. So we'll keep it simple and say lactose, the substrate for the metabolic pathway that we're talking about here. Lactose, a lactose-like molecule can bind allosterically, and when lactose binds, remember how these things work, it changes the conformation of the repressor. Rendering it inactive. Now, I just want to highlight this term inactive. For the repressor, we're going to say that when the repressor is bound, it is in its active form. So active repressor equals it is when it is bound to the operator. And therefore, its inactive form is when it is not bound. To the operator. So in the active form, this guy is sitting right here, docking at the operator region, preventing transcription. In its inactive form, it is not bound to the operator. And so I've just mentioned to you how this occurs. When lactose is present, lactose itself, right, this guy here, will come and bind allosterically, dock here. Again, it's allolactose, but you don't need to know that. So when lactose binds, it will change the conformation of this whole thing, causing this entire molecule, or causing the repressor to leave. So this binding here forces the repressor to move and makes it inactive. So now we have a repressor down here that is now changed in its conformation because it's bound allosterically to lactose. Okay, so this is the inactive form, for example. Now that we have the repressor removed, as long as the RNA polymerase is bound, that means as long as there's no glucose, now that this repressor is removed, you can then have gene expression. So you can have transcription, to produce a single mRNA transcript that has all three of these genes on it. And of course, translation can occur. What color? What color? Translation. And now we have our lactose permease and our beta galactosidase. And we have our third enzyme involved in galactose alterations. So there's enzyme 1, enzyme 2, enzyme 3, and they're correlated with these ones here, right? So enzyme 1 can go and let lactose in, enzyme 2 can help facilitate this conversion, enzyme 3 can go and do this thing. And that's how this whole thing works. So when lactose is present, the inducer, lactose will bind allosterically to the repressor and render the repressor inactive. The repressor, therefore, will not be able to bind to the operator and will be removed. When glucose is absent, then RNA polymerase will bind at the promoter region and transcribe the genes involved in this operon. Transcription and translation will produce the enzymes involved in lactose catabolism, right? And so lactose can be broken down to produce usable glucose that then can be converted to energy. So this is when lactose, so this is an example of an inducible operon where lactose is present and glucose is absent. So let's just write down like a few key notes here, right? For the lac operon, it is off or unable to bind when glucose is present and when lactose is absent. If either of these two conditions are met, then you will have everything turned off. It can be turned on only when both of these conditions are changed. When glucose is absent and lactose is present. Because, let's just indicate what each of these things mean. I'll try to color code this. Remember that when glucose is absent, that means that the RNA polymerase can bind at promoter at, oh geez, sorry, that's really ugly. That means RNA polymerase can bind at the promoter. The promoter, sorry, I just did that as a phosphate group, didn't I? At the promoter. Square. Okay, and lactose, the reason that lactose being present turns this thing on is because lactose or allolactose binds allosterically to the repressor, which removes from the operator. Right, so we have these two key regions, the regulatory region of this lac operon. When glucose is present, then we have the prevention of RNA polymerase from docking. When lactose is absent, we have the binding of the repressor. So when those both circumstances change, then we can have the RNA polymerase binding to the promoter region. And when lactose is present, then it can bind allosterically to the repressor and remove it from its docking station. Okay, I do have a question I want to ask you guys. Let me stop this for just a second. All right, so I wanted to give you a quick activity for this one based on what we just talked about here. I want you to work on answering this question. What is the fate of the lac operon when the following, when each of the following four circumstances are, I don't know how to say that right, under each of the following four circumstances. So when glucose is present and lactose is absent, when glucose is absent, lactose is absent, glucose is present, lactose is present, and glucose is absent, and lactose is present. So work through all four of those. And to answer these, indicate what's happening to the RNA polymerase and to the repressor, those two different factors that we just talked about as being affected by those two particular substrates. Okay. I'll save this one and upload this one too onto Canvas as an in-class discussion, because I think it's a good one for you to work on. And in our class discussion, we can look at this one in more detail. Okay. Sorry, I flew through a few of these things here. Let me back up. Back up. Okay. So here we go, this picture, let me just show you, we just talked about everything here, but let me show you what this illustration is indicating. So here at the top is showing you when this is repressed. We see this is repressed because the repressor is bound here. Now again, your book doesn't talk about glucose and its impact here, but remember that when glucose is present, you have no binding of the RNA polymerase, but when it's absent, then the RNA polymerase can bind to the promoter. Regardless of the binding of the promoter or the binding of the polymerase, you will have no transcription when the repressor is bound here. And so here's our three structural genes where this is indicating there's no synthesis of the enzymes here. Now when lactose is present, lactose or allolactose acts as the inducer of the entire operon by binding allosterically to the repressor. And this allosteric binding to the repressor causes a conformational shift in the repressor so that it is now inactive, right? So it's no longer able to bind to the operator. So it is removed from the operator. And once this is removed and you have the RNA polymerase binding to the promoter, then you can have activation of this operon and you have transcription of the three structural genes here, downstream translation of these genes into the three enzymes. And these three enzymes are involved in transportation of lactose inside and then digestion of this into usable sugars, right? And those are genes 1, 2, and 3 turning into enzymes 1, enzyme 2, and enzyme 3. Let's see, I'm not sure if this video will play, but let's see if it does. Nope, this video is not going to play for me here, sorry. So you can go in and watch this video on your own. This is just an illustration of the lac operon for, I'll check that next time, for understanding how this process works. But this is on our Canvas page, so just go and grab that video. You can pause it here and go watch that video for yourself. The two conditions, as just mentioned, that is required for the lac operon to be turned on are the presence of the substrate itself, so lactose, which acts as the inducer of the operon, and two, the absence of glucose, which in effect acts as an activator to allow the RNA polymerase to bind. So this graph, I think, is a great one for you guys to look at and try to understand what's happening metabolically. In fact, I challenge you to even stop it here and just try to figure out what this is talking about, and specifically figure out what substrate is being used for energy production under each of these phases in this growth, this population growth curve. But I'll explain it to you right now. This should look somewhat familiar to you. These are at least 3 of the stages of a bacterial population growth curve in a closed or a batch system. On the x-axis is the log of the number of viable cells. On the y-axis is the time of incubation. So at the very beginning of this particular population, you can see that glucose and lactose have both been added. Now, if you were to look at the population of bacteria in this first stage, which is called the lag stage. These organisms are adjusting to their environment. They are beginning their replication rate, but if you recall, the number of bacteria produced is just under a particular threshold to really be visualized on this log-based or semi-log-based scale. Nonetheless, if you were to look metabolically, like what's actually happening inside these cells, which of these two sugars are they using? Are they using both glucose and lactose? The answer is no. We've just talked about, and we've been talking about this for the last 2 chapters. When glucose is present, it will be used preferentially by that bacterial cell, either through fermentation or through respiration. Glucose is a key energy substrate or a key energy-rich molecule. And if you recall, the central metabolic pathways that we discussed in detail in chapter 7 are always expressed. All of the enzymes involved in that central metabolic pathway are constitutively expressed, so they're always on. So when glucose is present, it will be used preferentially. And we just discussed how lactose catabolism is suppressed unless it's actually needed, right? In this case, what is actually the fate of the lac operon? Well, let's just remember what each of these guys do. So when glucose is present, if you were to look at the fate of the lac operon right here, when glucose is present, glucose prevents the binding of the RNA polymerase. So RNA polymerase, and this is for the lac operon. RNA polymerase cannot bind. Sorry. RNA polymerase cannot bind to the promoter. Okay, so there's no binding of the polymerase because of the presence of glucose. Glucose prevents the binding indirectly again, but it still prevents the binding of that RNA polymerase. However, if you were to look at the fate of the repressor for the lac operon is what? If we see that lactose is present here, lactose binds allosterically to the repressor and causes a conformational change to remove it from the operon or the operator, right? So the repressor is inactive. That means it's not bound to the operator, right? But if you were to take these two into account, what is the fate of the lac operon? What does that mean in terms of whether there is use of this operon or not? Well, because of fact number 1, because there is no binding of the RNA polymerase, the lac operon is suppressed. You know what, let me change that to, can I erase that? Yep. Let me use a word that we have already used, let's say repressed, right? So the lac operon is repressed. Gosh. I will get this before the semester is over. The lac operon is repressed. Not in this case because of the repressor, but in this case because of the RNA polymerase unable to bind, right? No synthesis is occurring due to the fact that RNA polymerase cannot bind. So that is here at the beginning where glucose is present and lactose is also present. Although you don't have the repressor bound to the lac operon, you still have no expression of the genes involved in the lac operon because of the presence of glucose. So you'll only have glucose used preferentially, even though lactose is present in the media, it's just not going to be used. So in this growth phase, you'll see that the bacteria grow on glucose first, right? Glucose is used preferentially. So if you were to look metabolically, you'll find that glucose goes away essentially as it's being broken down, oxidized for the use or for the production of energy. Eventually, that population will reach, you guys remember what this is? Stationary. Right, this of course is exponential growth phase or logarithmic growth phase, and this is the stationary phase where the nutrients are being depleted. In this case, it's glucose that's being depleted here. But as glucose levels lower and the organisms have less glucose to consume, remember that the presence of glucose is the inhibitor in this case. So when glucose is gone, then you'll actually have the ability for RNA polymerase to bind. So at this point, when glucose is exhausted or gone, that means that you can have the RNA polymerase can bind to the promoter, right, on the lac operon. And if you recall, we already have lactose in the media, therefore we already have the inactivation of the repressor. So once we have the RNA polymerase binding to the promoter, this means we have induction. That means we have induction or synthesis of the genes involved, of the enzymes involved in bringing in more lactose and then breaking them down to produce usable glucose that can be used for energy. So now we have, at this point, we have a shift from using glucose to now using lactose. And once they shift over metabolically, they will begin an exponential growth phase again, this time on that alternate substrate, lactose, until lactose is exhausted. And they'll again enter stationary and eventually death phase. Okay, so again, this is a really great way to think about what's actually happening. In this case, glucose is present and lactose is present. In this case, glucose is absent and lactose is present. And so you can see what's happening on a metabolic level and genetically to the enzymes involved in lactose catabolism. Okay, here's a good discussion question. I am gonna review this here, I guess, and then I'll actually ask you this stuff as a lecture discussion question too. But let's look at this one here first. If an organism had a mutation in the LAC-I gene, this encodes for the lactose or the lac operon repressor, right? And if this mutation was in the active site such that it can no longer bind to its substrate, what effect would this have on the expression of the genes in the operon when glucose is absent and lactose is either present or absent? And actually, you know what I'm going to do is I'm going to not go over this one here yet, because I'll post this one during our discussion time from our Confer Zoom times, and then we'll have the answers to this one as we go through this. So I want you to think, though, as you're working on this problem, if the mutation occurs in the active site, I'll just kind of draw this out to get you started on this one. What we're talking about is the repressor of the operon, right? So here is our repressor. And it has an active site right here. And you have to first ask the question, what is the substrate that binds to the active site? If you recall, and going back to our whiteboard, we kind of drew this one out, right? We definitely drew this one out. If you go back to our repressor here, we already wrote it out. So here is the active site right here, and it binds to that region of the DNA. So this would be considered the substrate for this particular enzyme, for this repressor. And then remember, this is the allosteric region. So the question is asking, what if there was a mutation that occurred here? And we talked about the consequences of mutations in the active site in our last chapter on genetics, that if there's some mutation that made this region physically different, remember the basic rules, structure equals function for enzymes. So if the active site is mutated, then its function would be mutated, right, or changed. In this case, I already told you it cannot bind to its substrate. So what would be the effect of that mutation if it can't bind anymore on the operon when either of these two conditions is met, when glucose is absent and lactose is either present or lactose is absent? Try to review that one on your own, and then we'll discuss it in class. So that was enzyme induction, but there's also enzyme repression. So these would be biosynthesis pathways, right? Enzymes involved in the biosynthesis or anabolic reactions. Let's write that down. So these are for repression. These are anabolic. Metabolic pathways, anabolic pathways, or biosynthesis, right? Building up larger components. So normally these are in the on position. The cell wants to make more copies of itself, more and more, right? That's the whole MO of a bacterial cell, is just to continuously reproduce. So all of these different components building up, making larger lipids, for example, for the cell membrane, or making complex carbohydrates to go into the peptidoglycan layer, or making proteins for enzymes or for structural regions of the cell. All of those things need to be made at a constant basis. So they're actively expressed. All of these pathways, all of the enzymes involved in metabolic pathways that are for biosynthesis reaction. In this example, this is a picture of a repressible operon involved in arginine synthesis. Arginine is an amino acid. Every cell has a different capacity for which particular amino acids they can make. Of course, the others need to be provided as a nutritional component. The amino acids that can be made are synthesized unless the cell is either starving or the cell is provided that nutrient in its environment. Let's just say, for example, a neighboring cell died and lysed open, and suddenly all of their proteins became available for nutrient sources, and there was an abundance of arginine that came into the cell. And so in that case, this cell wants to turn off the synthesis of arginine since it became available in its environment. So when that product is no longer needed and the product goes back and turns off its own synthesis, remember we call this negative feedback regulation. Negative feedback regulation, where the product of the metabolic pathway turns off the enzymes involved. I don't know why it's crossing off, sorry guys, involved in that pathway. Okay, so it turns off the enzymes involved. In this case, we were talking about synthesis level regulation though. So it turns off the synthesis of the enzymes involved in this pathway. So here is under normal circumstances where this operon is on. Again, an operon is a collection of related genes, and we are talking about a metabolic pathway. So all of the enzymes involved in the synthesis, in this example, the synthesis of this amino acid arginine, all of these enzymes are regulated collectively or expressed collectively, right? So in this case, we have no inhibition. The RNA polymerase binds. It passes right over that regulatory region, the operator, and can transcribe all of the genes involved in this metabolic pathway. Here's the translation of these genes into proteins and the conversion of those polypeptides into enzymes. And now we have the ability for arginine synthesis. Now, again, when arginine or whatever this product builds up in the cell, because perhaps that nutrient has been provided, then that amino acid will actually go back. That amino acid, or that product, will go and bind allosterically, same exact concept, to a repressor, same concept as we just talked about. Under normal circumstances, the repressor is inactive, right? Which means it is not bound to the operator. But in this region, it's inactive when it's not actually regulated. It's inactive in its regular form. When it's transcribed and translated, it's normally resting in this inactive stage. To activate this, it actually needs that allosteric regulator, which binds allosterically, causes a conformational change, but in this case, the change renders that repressor active. Let's go down here where we have arginine accumulates. It binds allosterically to the repressor, and in this case, it renders that repressor active. So for repressible operons, the binding allosteric, the binding of allosteric regulator, which is the product of the pathway, renders the repressor active. Yeah, renders the repressor active. Let me just make that prettier for you. I'll just rewrite that. Okay, renders the repressor active. And remember, we said when the repressor is bound, it is called the active form. When it's not bound, it's called the inactive form. So for repressible operons or biosynthesis operons, ones involved in building up larger components, nutrients, whatever, when negative feedback regulation occurs and the product goes back and turns off the synthesis of the pathway itself, the enzymes involved in the pathway, the way it does so is by making the regulator, or making the repressor active, by binding allosterically and making the repressor active. Once that repressor becomes active, it sits down right on top of the operator, just like we saw before, and now RNA polymerase is blocked from transcription. There are some videos for this one too. You guys definitely go and watch the videos. And let's just do a practice question. Here's another question for you. A mutation that renders the regulator gene of a repressible operon inactive. Okay. Let's see. A mutation that renders the regulator gene of a repressible operon inactive would result in what? Yeah, actually, this is a good question. So a mutation that renders the regulator gene of a repressible operon inactive, well, it renders the repressor inactive, would result in what? Here are your four choices. Would you get continuous transcription of the structural genes controlled by that regulator or repressor? Would you have complete inhibition of transcription of the structural genes, irreversible binding of the repressor to the operator, or inactivation of the RNA polymerase? Again, we'll go over this during lecture. I want you to think about this stuff and see if you can answer that question. Okay, moving on. So that was looking at inducible and repressible operons and ways that the cell has to regulate gene expression. Now let's look at the physical changes that can occur to a cell during mutations or recombination events. So let's first start with mutations. What is a mutation? Well, it's simply an alteration of the nucleotide sequence in the chromosome. And usually this is what when we're concerned with mutations is when this is within an encoding region, so on a region where there's a gene. Now we're going to use different terms here, or we're going to use familiar terms you've heard of before in basic biology, but we're going to use them a little bit differently. So I'm going to talk to you about mutations on the genotypic scale, or the genetic changes, the physical alteration of the nucleotide, and then what the consequence of that genetic alteration is on the expressed polypeptide product, the protein product, right? And because you're changing it from a nucleic acid sequence to a physical protein, this will be the physical expression of that gene, or the phenotype. Now we know phenotypes, we talked about this in general biology as like hair color or eye color, right? The physical expression of your genetic information. Now, whereas this is kind of macro scale, your phenotype is also like what your enzymes look like, or what your lipids look like in your membranes, based on what those pathways are from the gene expression of the enzymes, right? So there's a lot of different ways to think of this term phenotype. So we're going to use these terms a little bit different from than you've seen before. Genotype is the actual nucleotide sequence. At the DNA level, specifically talking about genes. And whatever the expression of that nucleotide gene sequences will be the phenotype. and that is going to be your protein or your polypeptide product. Sequence of amino acids, right? And then downstream, your protein. So when we're talking about mutations, we'll say, what is the genotypic mutation? In other words, what specifically has happened to the nucleotide sequence? Have you had a single nucleotide switch from one type of nucleotide to the other? Have you had a big chunk deleted out, or a section put in, like an insertion, right? So that's physically talking about, or that's talking about at the genetic level, what's happened to the nucleotide sequence. And then I'll ask you, what's the consequence on the phenotype, or what is the phenotype? What is the phenotype change from that mutation? That means just looking at the amino acid sequence, the polypeptide product, has there been a change in that? And has the change been maybe a substitution, or no change at all, or an insertion or deletion? And then I'll ask you, what is the consequence on the final protein product? So these are 3 different ways to think about mutations, and I want you to be familiar with all of them, right? These other terms here, wild type versus mutant. When you have a wild type strain or the wild type sequence, that would be essentially what's in nature. That's the normal version, where you have the appropriate expression of that into a functional protein product, versus the mutant strain, right? That would be the mutated nucleotide sequence, which then could potentially have consequences downstream. So wild type versus mutant. And again, just so you're familiar with how we're going to discuss this stuff, wild type is WT and mutant is M or MT. Okay, wild type versus mutant strain. So there are different causes, as we've mentioned already. Spontaneous mutations occur regularly every replication event. Every time DNA is replicated from a parent cell to 2 daughter cells, normally those spontaneous mutations are in non-encoding regions of the DNA, so doesn't really have an impact, or has impacts on regulation, but not gene expression in particular. But sometimes those mutations can occur on the actual coding region and show some phenotypic change. Otherwise, we are talking about induced changes, and that would be during exposure of that bacterial cell to radiation, chemicals, or other types of environmental fluctuations. The genotypic mutations, again, we're using this term to talk about specifically what's happening at the nucleotide sequence level, at the DNA level. So here are the different terms you would use to describe specifically at the DNA level what's happening to the nucleotide sequence. If there are multiple nucleotide changes, so you have an insertion or deletion, right? Insertions or deletions. So adding or taking out nucleotides, this would just be insertion or deletion is what you would actually state. And you could state how many nucleotides have been inserted or deleted. For example, insertion of 1 single nucleotide or insertion of 5 nucleotides, whatever that might be. Alternately, you're talking about single point mutations. That would be additions, deletions for single point mutations or substitutions. So if you were to be asked, for example, in this picture here, where we have our parental DNA, there's a mismatch that has been incorporated as a mutation. And this is the mutant sequence versus the wild type sequence. You can see that G's and C's would be here and A's and T's are here instead. This is an example of a single point mutation because there's only a single nucleotide change, and it's a substitution where 1 nucleotide has been substituted to another. Now, it's also important to note in this picture here, spontaneous mutations that occur during the process of DNA replication, usually these mutations are fixed. And I'll talk about that in just a few minutes about this DNA repair mechanism we have. So usually, spontaneous mutations that occur during DNA replication like this are fixed by machinery, by repair machinery. But if they are not fixed, then look, this is actually not called a mutation. This is called a mismatch at this point right here. Let me just write that down for you. So when you have an accidental or spontaneous mutational event that occurs during DNA replication, at this point here, this is only called a mismatch. Because you have a mismatch in the nucleotides, right? G's and T's do not fit very well together, essentially. And this usually does cause a physical change to the DNA, a little bulge in this thing. making it not fold very properly. And usually repair mechanisms take care of this. So usually repair mechanisms fix this, but if they don't, then upon the second replication event, then this template, right, this template will now act as, or this strand here will now act as a new template, and now you'll have an incorporation of that mismatch as a mutation. So when you actually have both of these nucleotides bound correctly in the correct complementary base pairing, but this base pairing is actually different than what it's supposed to be, now we call this a mutation. So on spontaneous mutations that occur during DNA replication, essentially the first step is just a mismatch. If it's not fixed, then upon the second replication event where that mismatched, that aberrant incorporation of that nucleotide, when that becomes a template strand, now you're going to have that new strand have a mutation in it because of that alteration. I'm going to pause here for just a second. Okay, I've got some starving people. Let's go back and finish this one. Okay, so let's look at what the consequences of these genotypic mutations are on the polypeptide product. Okay, so what are those phenotypic changes? When there's a change to the genotype or the nucleic acid sequence, this will lead to a change in the phenotype or the polypeptide, right? The sequence of of amino acids as a polypeptide product. And of course, downstream, this can have consequences to the protein itself. These are the terms, the new vocabulary terms for you: missense, nonsense, frame shift, silent and back mutation. Let's just look at each of these things in detail. When there is a change to the nucleotide sequence that leads to a substitution from one amino acid or one codon that encodes for one amino acid to another codon that encodes for a different amino acid, this change in amino acids from one type to another is just called a missense mutation. This is just a substitution of one type of amino acid to another. You can look this stuff up by going to your amino acid codon chart or mRNA codon chart or tRNA codon chart, and you can look up specifically GAA. You can look this up and you would find it encodes for a glutamine versus GUA. This one now encodes for a valine. This picture that we're showing you here, this is actually a true mutation that has occurred in a large percentage of African American population. This is sickle cell anemia. Where this is the physical single nucleotide change. So again, if you were to talk about genotypic and phenotypic mutation, I would say this is a single point mutation substitution, where a single nucleotide has been substituted for another. And the consequence on the polypeptide product is a missense mutation, where an amino acid has been substituted for another. Downstream, if you know anything about this, sickle cell means it actually changes the conformation of that cell, making it, or that hemoglobin molecule actually changes the cell itself, and it causes anemia because of the reduced ability for that hemoglobin to pick up oxygen. Well, whatever. Anemia is about red blood cells, but this also reduces oxygen saturation here. So, whoops. So for this particular change, you also can indicate not only the phenotypic change, which would be missense mutation, change in the amino acid from one type to the other, but downstream, the consequence on the protein product would be creating a less or non-functional protein product. The next example is a nonsense mutation. This is when an amino acid is changed to a stop codon. And remember, there are 3 different stop codons that are encoded by 3 different three different stop codons that we have on our genetic code. And in this example, you'll see that that cytosine, there's a replacement of a single nucleotide. So genetically, we're saying this is a single point substitution mutation from a cytosine to a thymine. If you were to look this up, CAG in DNA downstream would encode for a glutamine. PAG in DNA downstream would encode for a stop codon. Again, you could just do that transcription translation yourself, go look this up in your codon chart, and you would find that that's true. So a nonsense mutation is where an amino acid changes into a stop codon. The consequence would be a truncation of that polypeptide product. Truncation means you're just chopping it off right there. Usually, almost always, this results in the loss of function. So that protein just won't work. You just don't have the rest of that protein. The very few examples where that might not be the case is if this occurs, right down at the very end of that sequence where that stop doesn't really make a significant impact. But almost always, this leads to a complete loss of function. A silent mutation, this has to do with the redundancy of our codon chart. If you remember that term, that means that there are sometimes up to 4 different codons that can encode for the exact same amino acid based on that tRNA anticodon, if you recall. So this silent mutation is where there is a mutational event. So here's an example again of a single point mutation where that cytosine has been changed to a guanine, and this leads to a substitution mutation genetically. But if you were to look at this mRNA sequence, UUG, and this mRNA sequence or codon UUC, you would find that both of these encodes for phenylalanine. So the polypeptide product is not changed. So this is a mutation, right? It's a genetic mutation, but it encodes for no change in the amino acid product, or it encodes for the same amino acid. So we call this a silent mutation. The consequences downstream on the protein, there are none, right? This has no change in the protein function. A back mutation is one that's kind of tricky to describe, but essentially this is a double mutational event. So we have these regions of DNA, we have them, prokaryotes do, called mutational hotspots. And it's due to the folding of our chromosomes or prokaryotic chromosomes, where sometimes there are more exposed regions that are just more subject to mutational events. So let's say this was the normal wild type sequence. There happened to be a mutational event that conferred a substitution from T to A. And then a back mutation is when at the exact same site on the gene, that mutational event occurred again, reverting the mutation back to the wild type form. Right? So this is called a back mutation. And the consequence would be reverting a mutant to a wild type form again. And the last one here would be a frame shift mutation. This one goes back to our discussion of frame shifts, where we talked about how if you change your reading frame by grouping your codons at the first nucleotide or the second nucleotide or the third, gives you a completely different reading frame, right? Or different codon sequence, which then leads to a completely different polypeptide product. So a frame shift mutation is when you have an insertion or deletion event of any number of nucleotides except for groups of 3. If you imagine, if you take out or put in groups of 3 nucleotides, essentially you're just going to be disrupting that polypeptide sequence by putting in an extra amino acid or taking out an amino acid, but you have no change on the reading frame. But a frame shift is when you actually shift that whole reading frame so that there is a consequence on the entire reading or the reading of that mRNA transcript to produce a totally different polypeptide sequence. Sometimes that reading frame shift can cause an immediate nonsense mutation. So you have a stop codon that truncates everything, completely eliminates the protein product. Here's an example where an extra U was put in here. So whereas the next codon would be AAG, if there's an insertion event here, then the next codon would actually be, in this example, UAA. Here it was AAG, it's still there, but you can see that you don't read it anymore as AAG because now this is our codon and this encodes for a stop. So sometimes the consequence can be immediate nonsense. Sometimes there can be excessive missense mutations that occur. For example, in this case, that U has been taken out, which shifts everything over. So instead of UUU being the next codon here, it would be UUG, and this is the leucine. The next would be GCU, which is an alanine. And so you can see that the polypeptide product becomes totally different. If again, there's an insertion or deletion of 3 nucleotides or any sets of 3, it doesn't actually change the reading frame. It just adds or removes an amino acid altogether. The consequence of frame shift mutations are almost always a loss of function for that protein product. Okay, this is a set of discussion questions. These each are examples of different mutations that result in different phenotypic alterations. So you'll have a missense, nonsense, frame shift, and silent mutation in all 4 of these. This will be one of your in-class discussion questions. I'll do this together with you when we get to this portion of our lecture together. You can see how to do these things, but this is a great way for you to practice transcription, translation, and then understand the consequences of each of these mutations on the protein products. So I'm going to pause the video here and I'll pick back up with a second video for our very last section on DNA replication, DNA repair, and genetic exchange.